Lab number 3 is going to be about physical transport. And the way we're going to run this lab is as follows. So we're going to go over the important terms. It's not too much. So if you can kind of see, it's not too many terms. I'm also going to give you an introduction into the various experiments that you would be doing on site. Now, some of them you can do something very similar at home, and I'm going to give instructions of how to do that. And others, they will be shown to you as demo videos where somebody is doing that experiment, and it's a YouTube video, and those links are already on your Canvas website. So, but let's get started with the important terms. And the first 3 here about the states of matter, which I'm sure you are familiar with. Matter can exist in a solid state, in a liquid state, in a gas state. And the difference is, so most matter can transition from solid to liquid or gas. Some will transition like directly from solid to gas, for example, like dry ice. But most matter transitions from solid to liquid to gas, depending on temperature. And the problem is that the in a gas state, of course, the molecules are not as close together, they're not as packed as they would be in a liquid, or in most extreme cases, solid. So the difference is molecular movement, and molecular movement is determined by heat. So the more heat something has, the more molecular movement, the more bounciness is there among these molecules, and the more they are sort of bouncing apart from one another, and then they transition among these 3 states of matter. An important term that you need to know is rate, and that's always change per time. So it's sort of something like diffusing a certain distance per whatever time increment. And time increments, popular ones are per second, per minute, per hour, per day. So depending on what the material is. But keep in mind that rate is always something is a change per time. The next 2 terms, kinetic energy versus potential energy. Kinetic energy, if you were the term kinesiology, kinetics is movement. So that is energy being used. Think of you driving a car, or you riding a bike, or you running, or a log of wood burning. It's energy in motion, energy being used. Potential energy is, it's stored energy. So you're not using it right now. It's the log of wood in your fireplace that you have and that you're not using right now. It's the car with the gas in it in the garage. It's you on top of a hill with your bicycle, but you're just standing there. And once you are rolling downhill, well, then you transition to kinetic energy. So potential energy is energy stored. Kinetic energy is energy used. Brownian movement or motion energy, that is what I was just earlier saying about. Brownian movement refers to these states of matter. So the more heat something has, the more Brownian movement or molecular movement something has. And the hotter something is, the more energy it contains, the more bounciness is in these molecules, and the more they want to have space and bounce apart from one another. And therefore, they transition from solid to liquid to gas. And you know that, think about cold water. How damaging is cold water? Not that damaging, but hot water is quite damaging. So the only difference is temperature, is still water. But how is it that hot water is such an aggressive kind of damaging substance versus cold water is not damaging at all? So the answer is it's in the energy, it's in the collisions that the matter creates as these molecules are bouncing around one another. Okay, the next terms are all referring to various transport mechanisms, and the ones that are listed right here, they are all passive transport mechanisms. Physical transport is kind of a big deal in physiology because we're always talking about stuff moving in and out of cells, and we have these fluid compartments, extra and intracellular fluid compartments and whatnot. So stuff is always being transported back and forth in the human body, within cells, and in and out of cells. And so transport is a big deal thing. Now, because there's so much transport that's necessary, cells, they don't like to spend that much energy. So whenever possible, they like to use something that is energetically for free. And that means we're using passive transport processes. So I like to create a little table here at this point to show strictly separate the transport processes a bit into active and passive transport processes, because I think that will make the most sense to you, and it's the easiest to sort of remember about these different transport mechanisms. So I'm going to stop the recording here and just do this to create that table. Okay, so here's the table, and it's sort of my own spin on these transport mechanisms. I think it makes a lot of sense to strictly separate into passive and active transport processes as we talk about this. And so let's take a look first at the passive transport processes. The beauty of that is that you do not need to put in any energy. So there's no ATP requirement. The only requirement you have to have is you must have a concentration gradient. If you don't have a concentration gradient, these passive transport processes will not work. Examples of passive transport processes include simple and facilitated diffusion, and then the all-important movement of water across a semi-permeable membrane that's known as osmosis. Also filtration and dialysis are a good example of passive transport processes. On the active side, well, we're going to have to put in energy. We're going to have to put in sometimes a lot of energy. So ATP is required, but you don't need a concentration gradient. In fact, a lot of times we're establishing a concentration gradient. So we're putting energy into the system. Examples for that include all the ion pumps. A good example here on this end is the sodium-potassium pump. That is very important. That one alone, that one protein that pumps sodium to the outside of biological membranes and potassium in the inside of the cells, that one pump keeps your nervous system alive and functioning. And it requires about 1/3 of your basic metabolic energy. That's a lot of energy. That's a lot of ATP, but you gotta have it. Then also bulk transport is active and requires energy that would include exocytosis, such as the release of a neurotransmitter at a synapse, and also endocytosis. That's when cells take something in, sort of enclose it in a transport vesicle. If that particle is solid, then we call it phagocytosis, which is literally cell eating. If what the cell is taking in is liquid, then we call it pinocytosis. Okay, so this is sort of your table that will help you, or should help you, orient the different and categorize the different transport mechanisms. For the sake of this lab, we will be staying entirely on the passive side. So there are no experiments in here, or even in our on-site lab, we would not be doing any experiments along the lines of active transport. So I will cover all of these terms right here. So let's move on to hypo, iso, and hypertonicity. Tonic means strength. And basically what you're doing here is you're comparing the ionic strengths, or the amount of dissolved particles. You're comparing the two solutions to one another. If they have the same amount of dissolved particles, we call them isotonic. That means they have the same strength. It's basically the strength to pull water and as a form, with osmosis. Now, hypotonicity or hypotonic means that my reference solution, the one I'm looking at, has less and fewer dissolved particles than the one I'm comparing it to. So if you're comparing distilled water to ocean water, then the distilled water has fewer dissolved particles. In fact, it has 0 dissolved particles. So the distilled water is hypotonic compared to the ocean water, which has a lot more dissolved particles. If I'm comparing ocean water to distilled water, well, then the ocean water is hypertonic compared to the distilled water, because the ocean water, again, has more dissolved particles compared to the distilled water. And if the two solutions have the same amount of dissolved particles, then we call them isotonic. There are a couple of experiments along those lines in this lab where you're taking a look at cells, what happens to cells when you're placing them into isotonic saline, isotonic, that is, to the cell. And there you will see that's an equal exchange, and in terms of volume change, nothing will happen. If you're putting cells into hypotonic environment, let's say you're putting red blood cells into distilled water, you will find that they quickly will swell up and burst. And that is called lysis. So here we have the term lysis. So when a cell bursts due to swelling up and water intake, and then it bursts, and then that's lysis. And that happens to animal cells a lot because they don't have cell walls. Plant cells are protected by their cell walls, and so are bacterial cells, but our poor animal cells, they can't withstand that. So if you're placing red blood cells into distilled water, they will quickly burst, and there's a video that will show you that. It's actually quite good because it shows you under the microscope how these red blood cells burst, which is something we always have a hard time on site to demonstrate that because students tend to use too many red blood cells, but in that video is quite good. If you're placing cells into a hypertonic environment, and that means the cells have fewer dissolved particles than the solution that they're placed into, then they tend to lose water because water will go down a concentration gradient. And if the cells have more water than the outside medium, then water is going to leave the cell and dilute sort of this outside medium. So if you're putting your cells, red blood cells, let's say, into ocean water or 5 or 10% saline, which is what we have in the lab, then they quickly will shrivel up, they will lose water, and that process is called crenation. So when these cells shrink due to water loss, it's called crenation. Couple more terms here. A solution is composed of a solvent and a solute. In our case, the solvent is always water, and the solute could be various things. We have sugar molecules, molasses, for example. We have salt, of course, as a solute, and other things. But keep in mind, a solution is composed of a solvent, in our case, always water, and a solute, whatever you're dissolving in that water. As part of the lab, you would have done a simple filtration experiment. We have a mixture of a crystalloid, a colloid, and a mechanical suspension. For the crystalloid, we're giving you ferric chloride. It's like a yellow salt. The colloid is starch. There's also, they are larger particles than a crystalloid, but they will not settle out. They're too small to settle out, but they're too big to go fully in solution, so they make the solution kind of cloudy. It's like cells would represent a colloid as well. So if you're looking at blood, it's not transparent because the blood cells are suspended. They're colloid in the plasma in this case. And starch is a good colloid too. The starch particles stay in solution, but they add turbidity to the solution. And then a mechanical suspension, we actually have charcoal. We have charcoal that we use, and that one settles actually out. Or think of sand, something larger. It will settle out. It's a mechanical suspension. Ways to measure concentration is either percent by mass or percent by volume. That's what we usually do. So we have, let's say, 5% salt solution. That means we dissolve 5 grams of salt in 100 milliliters of water. You can also use molarity. Chemistry people like that much more, but in biology we usually have percent by mass or percent by volume. That's how we indicate concentration. And concentration gradients, that's of course, that's what you must have in order to do any kind of passive transport process. And indicators, we have indicators that demonstrate the presence of something, and I will write that down for you real quick. Hang on. Okay, so here are our indicator reactions. We demonstrate the presence, and so that's a positive test. The presence of starch with iodine. So if you add a drop of iodine solution to starch, it turns dark blue, and it's a very obvious reaction. You can't miss it. Sodium chloride and silver nitrate will form a insoluble silver chloride precipitation, a cloudy precipitation, and that is used to demonstrate the presence of salt, chloride to be specific, and the presence of glucose and protein in this experiment is always demonstrated with the use of heuristics, where just simply use a dipstick, and if it changes color, then you know there's protein or glucose present. So moving on here to the actual experiments. On page 40, you'll find experiment A. You won't be able to do that, and it usually doesn't work that well anyway, so no big deal. Experiment B, please watch a video. It demonstrates exactly that experiment. Experiment C, filtration. You can do that easily at home. You can use either coffee or tea to filter, and that should work. Experiment D, please watch a video. And for experiment E, also demonstrated on a video. So please go on Canvas and go through these videos that I posted, and you should be fine with that.